Welcome to the biannual MACAN meeting. For those of you who I have not met yet, my name is Janet Reamer. I am the Ocean Sciences Director, or Ocean Sciences Program Manager, for the Mid-Atlantic Regional Council on the Ocean. And today with MARICUS, the Mid-Atlantic Regional Association Coastal Ocean Observing Systems. We were just talking about acronyms. We'd like to welcome you all to our workshop today, and want to welcome a lot of new faces. And I myself have only been with Marco for about 6 months, so this is really exciting for me. I haven't been to a MACAN meeting in several years, and it's our first time back in person in a while. So we're all really happy that everyone was able to come today, and we have a day and a half of some great research updates, stakeholder outreach, basically everything ocean acidification and marine carbon dioxide removal that you can think of, we have tried to cover over the next few days. So just a few meeting notes. Well, actually the first thing I want to say is that you're probably all very familiar with the one of the activities that MACAN has been participating in over the last few years, which is the, I'm probably going to say this wrong, but don't worry, I'll get corrected later, the Coastal Communities National Vulnerability Assessment that was conducted by the IWGOA. You'll get more information about that later, but it was finally released today, which we're all super excited about because all they can support really hard to support this effort, and we're going to hear more about it, and it's really awesome. If you are not on the Wi-Fi network, and you want to be, is the visitor network. There is a water refill station, so I see so many water bottles on the table, which is totally awesome. Thank you. And yes, you see all of this video equipment and AV equipment. This meeting is being recorded, so if you do not want to have your talk on the recording, which is going through the MACAN website, please let either myself or Kirsten know so that we can make a note of that, and it will be edited out. Additional AV that you'll be seeing today is through Greenfin Productions, which MACAN was funded to do some educational videos. It is completely optional. You can talk to Dave, raise your hand, that's Dave right there. If you're interested in participating in one of the interviews, we have some interview questions. We did share them before the meeting, but if you are interested in participating, we'll share them again with you, and Dave can also give you more information about that. Okay. Oh, sorry. Thank you. So for those of you who may not have been to a MACAN meeting before, or we may have contacted you somewhat out of the blue, just a little bit about what MACAN does. We're a nexus of scientists, federal, tribal, and state agency representatives and resource managers, also industry partners in the fisheries and aquaculture industry, and we all come together to coordinate and guide regional observation, research, and modeling of ocean and coastal acidification. Overall, the CANs in general have pretty much the same goals and missions, which are to better understand all of the drivers of coastal and ocean acidification, predict the consequences on marine resources, coordinate best practices for monitoring and data collection, and also help with stakeholder education and outreach so that someday we can hopefully mitigate this problem. One of the things I was asked earlier today is how do you explain ocean acidification in 10 seconds or less, not using jargon. Let's just say that I practiced it in front of the mirror about 10 times and still flunked it. So it is not easy to do. But using jargon, acidification is a combination of water and carbon dioxide and the carbonate ion, which form 2-bicarbonate, which impedes the consumption of, the consumption of carbonate ions then impedes the calcification process, which is important for all shellfish and corals and has impacts especially on early life stages. So if you are here with us today and are learning about acidification, a great place to start, of course, is the MACAN website. So just to give you a little overview of what MACAN has been working on over the last year and into the 2024 fiscal year, because of our funding from NOAA's Ocean Acidification Program and through our bipartisan infrastructure law funding, we have quite a few projects going on, some of which I'm still mixing up and confusing. So if I misidentify your project, then that's on me. So earlier this year, we conducted our webinar series, which consisted of 4 stakeholder-oriented webinars. We are very involved in cross-CAN coordination, especially with the East Coast CANs, and we're very happy to say that all East Coast CANs are represented in this room today. So that's pretty exciting. I'm not sure that that's happened at a regional CAN meeting yet, so very proud of that. We, over the last, I'm going to go with 2 years because I'm pretty sure it's been more than that, though. We have helped inform NOAA's Coastal Communities Vulnerability Assessment. MACAN has participated in the production and release of a curriculum in coordination with the Mid-Atlantic National Estuarine Research Reserve programs, specifically the one in Chesapeake Bay, correct? Yeah, all of them, sorry. But I want to call out Chesapeake Bay as well because it was kind of, I think it originated down in Chesapeake, right? Okay. One of the projects that we're just starting to take on is educational video shorts and our social media. So if you are on Instagram or Facebook, I think it's mid-ACAN, thank you, mid-ACAN, and please go ahead for both of them, follow, like, share. We are undergoing a full website refresh and redesign, probably finishing up later this year. So when that happens, obviously we'll have a big email celebration and send that all out to you guys. We're just starting an acidification hotspots layer, which is going to be compiled over the next about year to year and a half, and will go up onto our the Mid-Atlantic Ocean Data Portal and Maricosta's Oceans Map. So we'll be reaching out through our steering committee and obviously Kirsten and myself over the next year or so to kind of get some guidance and help with that project. We just finished up a refresh of the monitoring inventory. So some of you may be familiar with the fact that back in about 2017, there was the initial layer that was displayed on the portal that is basically just gives locations of where ocean acidification is being monitored. So through any 1, 2, 3, or 4 of the carbonate system variables, and that layer has been updated with all of the new sites and data that's being collected. And our final one of our projects that we're going to touch on over the next few days is a MACAN Workforce Development Fellowship. There are a couple of little flyers that are in the acrylic holders that you'll see around. The deadline for that fellowship is going to be October 1st, so we're kind of coming up on that deadline. But the Workforce Development Fellowship is specifically directed towards underrepresented and underserved communities. It's going to be a year-long fellowship that will expose the selected student, either a 2-year college, 4-year college, or graduate student level. We'll be mentoring them and exposing them to all the different aspects of ocean acidification careers, from planning to science, communication, and education. So if you are interested, as well as being part of that mentorship program from the mentor side, we would love to hear from you. We're trying to get as broad a range of career opportunities There will be travel involved for the student, and please feel free to take the information back to any of your students. Like you can scan the QR code on any of the acrylic displays. So from there, I'm going to just run you through a little bit of today's agenda. We're going to have Kirsten give us the tribal land acknowledgement, and then we'll run straight into our first session, assessing vulnerability in mid-Atlantic species, ecosystems, and industries. And then we're going to go right into session 2, new initiatives in community outreach and engagement. We'll have a brief coffee break, which I think is sponsored through Maricruz? I don't know. Maybe. Let's go with that. Thank you, Maricruz. And then our final session of the day will be updates on federal and state OA initiatives and diversity, equity, inclusion, justice, and accessibility considerations. We'll have speakers for the panels, panel sessions, come up and just take a seat at the front table, and then you'll be able to give your presentation, and then we'll ask you to sit back down, and then we'll have questions and answers at the end of each of the sessions. So, that, here's it.
Thanks, Janet. It's nice to be here today. For those of you who don't know me, I'm Kirsten Wakefield. I'm a stakeholder engagement specialist with Maricruz, and I'm also the co-coordinator along with Janet for MACAN. We've been asked to give a tribal land acknowledgement today, so we'll start with that. We take this moment to acknowledge where we are today at the Institute of Marine and Environmental Technology in Baltimore, Maryland. We honor the land on which we stand, the traditional land of the Susquehannock, people of the Muddy River, and the Piscataway, people where the rivers meet. We pay respect to elders both past and present, and we take a moment of purposeful silence to acknowledge the history, path, pain, and triumph of ancestral stewards of this land and indigenous nations and peoples across the United States. We respect the ancestors and their experience and move in the direction of intentional collaborative stewardship today. And we thank the New York State Empire Fellows, Nicole Jean Christian and Chelsea DeJesus for their guidance in helping to write this acknowledgement. All right, so for our first session today, assessing vulnerability in mid-Atlantic species, ecosystems, and industries, we'll have 4 speakers. The first is Shannon Misek from the NOAA Northeast Fisheries Science Center. Our second speaker today will be Jim LeChant, who's a social scientist with NOAA. And our third speaker today will be Emily Revest. She's at Virginia Institute of Marine Science. And we'll conclude the session today with Kathy Gervitz, who's at St. Mary's College of Maryland. So as Janet mentioned, if you all come up and give your presentation and then have a seat until the question and answer at the end, we'll go ahead and do that. All right? Thanks so much.
Figure out technology. Thank you for the opportunity to speak here today. I'm going to be talking on a project I've been working on. It's multidisciplinary with multiple people involved on it, looking at some of the chemistry in the area, looking at the ecological effects on sea scallops, and looking on the social effects with the industry. So it's a big collaborative project that we've been working on together. I'm going to focus a lot on the physiology of the sea scallop. So to begin with, the sea scallop is located from Canada all the way down to Cape Hatteras. It is increased in revenue in the United States. It's over $500 million every year in harvest. And as you can see from this figure, since 1958 to recently, the amount of sea scallops being removed is increasing. More of the industry is switching from other revenues to the scallop industry. So that's been happening in the Northeast United States. The sea scallop, if you're not familiar with it, has a long lifespan. It can live up to 20 years. It is well managed by the industry, and it takes about 4 to 5 years before it will reach harvest size. It reproduces in about 3 years, and when it reproduces, it occupies different water masses. So it'll reproduce and it will go up into the water column as a larvae and become planktonic. And then about 40 days later, 40 to 50 days later, it'll start to settle on the bottom and become benthic. So it has two life stages that means it's experiencing a very dynamic environment when it is first, when spawning first occurs. And so we need to keep that into account when we're looking at the sea scallop. So this is recently a figure that just went into the State of the Ecosystem report for NOAA fisheries. And this is work that Grace Saba is doing with the gliders to look at what's going on with the aragonite saturation state during the summer. And as you can see, it varies up and down the east coast in the habitat where sea scallops are located. Levels vary as high as 2.2 to as low as 0.8. And there has been a region identified off the coast of New Jersey right now where the aragonite saturation state has dropped below what is considered sensitive for the sea scallop. That recently just went into the State of the Ecosystem report. The fisheries has seen a lot of changes over the last 30 to 40 years because of it was declared overfished in around 1997. When it was declared overfished, management came in, they started doing rotational area management, so that resulted in a peak biomass. The idea between the management and the rotational closures, which officially began in 2004, is if you let the small, fast-growing sea scallops stay there, you protect them, and you can increase sea scallop meat and boost yield. This was a collaborative work between the industry and NOAA fisheries to make sure that we could keep recruitment in the fisheries going. And recently there's been a shift in biomass, and as you can see from this, it appears that more of the industry is being harvested in the Gulf of Maine versus in the Mid-Atlantic. So the fisheries is seeing changes. Is it because of acidification? Is it because of warming? Is it both? Is it because of the rotational? Those are all questions that we are trying to address. So this project has an integrative approach where we determine what the historic conditions of the ocean were, what the projected future conditions in their habitats going to be doing, combined it with the physiological response of experiments that we get to predict how the sea scallop populations may move. We also assess what's going on historically with the industry to figure out what the industry is going to do. And we just keep on doing this in this project. And I'm going to show you, as I said, scallop physiology, because that's what I've worked on. So as I said, the sea scallop lifespan has two phases. There's the pelagic phase and the benthic phase. Sea scallops, if you've ever tried to raise them, are notoriously difficult. You get to about day 21 and then they die on you because you're trying to do it in the laboratory. So we're going to focus on what is known as the juvenile spat phase. The sea scallops we got in were from the Gulf of Maine. They ranged in size from 10 millimeters to about 30 millimeters. And what we did is we exposed them to various ocean acidification conditions. And our thought behind this was we wanted to be able to generate this dynamic energy budget model that would allow us to see how they respond to CO2 and changes in temperature. So if you can understand the physiological process, they ingest food, and that allows them to grow, and then it costs them energy to digest that food, and it costs them oxygen, so that they have an oxygen demand, and they also have an excretion demand. So if we can look and see if how they're eating is changing, or how they're respiring, or how they're excreting based on CO2, we can then put it in this model and look at how they vary their growth based on food availability, based on temperature changes, and based on carbon dioxide. That then tells us whether or not they're putting the energy into making meat, or reproduction. So what we did is the juvenile sea scallop experiment, and this has been published in PLOS Climate, and we held 3 CO2 conditions. We had low, medium, and high. They ranged from current conditions up to 1,200 microatmospheres. We fed all of them algae to make sure food was not limited, so they got supplemented. And what we were trying to do is measure those physiological rates of respiration, feeding, and excretion. But we were doing it during a natural temperature drop, because temperature isn't constant in the ocean. It varies quite a bit. So this is a natural temperature drop like they would experience in George's Bay. And we were trying to see what would happen. Would any of their physiological rates change? How do they change? Is the temperature drop more? What is the role of the temperature drop, and what is the role of the CO2? So this was our carbonate chemistry, and as you can see, the treatments they did drop in temperature just like you would in the field, and they were consistent between all 3 treatments. And if you're interested, those were the concentrations we saw for pH, DIC, and PCO2 and calcite. So this summarized all the physiological results that we had from the experiment. And what was interesting is we looked at just what was going on with carbon dioxide. We saw that feeding decreased as you increase CO2. They were less efficient at feeding when we increased the CO2. But we saw no changes in respiration and no changes in excretion. Carbon dioxide didn't affect those metabolic processes. If we turn to temperature, we got a different story. Temperature was also caused the change in feeding, with maximum feeding around 9.4 degrees C. This max feeding agrees with the literature. If you go into the literature from other experiments, it varies between 8 degrees C to 12 degrees C where you have max feeding. So we were seeing that too. Respiration increased with temperature, which is very common for sea scallops too. So we were seeing that with our experiment too. And so did excretion. What is interesting between both of these is that there is an interaction between carbon dioxide and temperature for feeding behavior and for excretion rates and the O to N ratio. If you're not familiar with the O to N ratio, you can take the respiration rate and the excretion rate, and you can calculate the amount of oxygen to nitrogen. And the importance of that is it gives you a proxy of whether or not they're degrading proteins or lipids and carbohydrates. When they degrade lipids and carbohydrates, you would expect an O to N ratio anywhere above 15 to 20. It depends on the bivalve. And it could go up to 100 on some bivalves. As you get closer and closer, lower than 15, it means you're digesting more protein. For scallops, the threshold of 100% protein degradation is 8.9. So keep that in mind when I show some figures here. So that's really important, and one of the reasons they switch to protein catabolism, if they can, is because even though it generates ammonia, it actually allows them to maintain their internal pH. And that's why they like to do it. And it gives them more energy so that they can continue to grow. So if you actually look at the interaction, which was the really interesting part to me about this study, is what we saw is a thermal performance change because of increased carbon dioxide. So when we were at current levels, we get this, for assimilation of the food, we get this nice U-shaped response. So at maximum temperature, they're assimilating food like they should. When temperature gets really cold, they slow down, and when temperature gets really high, they can't feed either. That's typical bivalve response. But what happens as we increase the CO2, that thermal threshold moved. Now they no longer had this nice U response, and they were feeding very similar at 6 degrees and 7 degrees. So they weren't able to switch as quickly as they could at the lower CO2. And then on top of it, what we ended up seeing is the excretion rate ended up changing, whereas we had our lowest ammonia excretion at around 7.4, so they weren't doing a lot of protein catabolism then. But then as you increase CO2, it just kept on increasing the amount of ammonia being excreted. And this resulted in our O to N ratios being basically the same. They were, when we ended up being at low CO2, we were going between protein catabolism and carbohydrate and lipid catabolism. But when we got to high CO2, we were only able to observe that they were pretty much using protein catabolism. And we believe this is because they're trying to maintain their internal pH. And what this has on reproduction, we're not really sure right now what that will translate to. For scallops, one of the reasons they can switch is they almost always have protein catabolism activated in order to swim. Have you ever seen a swimming scallop? They're really fun to watch. They need protein catabolism. They always keep it activated so they can swim and evade predators. So that's what we found interesting in this, is that it appears that as we increase CO2 in the environment, there might be a change in the thermal preference. This data is then being put into a dynamic energy budget model that Hallie Berger from UConn is working on right now. So what she has is CO2 is affecting the food that they're ingesting and the reserves, and that then goes into the reserves for structure or reproduction. Temperature affected everything, affected food intake, respiration, and excretion. And then it will be linked to a regional ocean model output, so we can see what is going on in the habitat of sea scallops. So this is preliminary results that Hallie has modeled, and as you can see, as you move forward in Georges Bank from 1980 to 2000, and then the year 2080, there's a decrease in shell length of sea scallops. And it takes them longer to hit the length for harvest size. So that's an important aspect that she's finding with the model as she links it to some of these climate variabilities. So this model will continue to be developed because she'll also look at what's going on with reproduction in the model and generate maps to predict growth rates, how times to harvest would change, and if there would be any potential changes in recruitment, including changing in timing. They may end up spawning earlier versus duration. They may spawn less throughout the season. So those will be some of the things that will be coming out of Hallie's work. The other thing is, in the middle of this project, we found out that NOAA fisheries and fins have a lot of shells in storage. And there have been over 3,000 shells measured to date to look at if there has been a difference in shell thickness, because one of the parameters that's often affected with these experiments is the shell thickness is affected, or shell length. And so Hallie is going through that archive and measuring every single shell, and they're doing analysis to see what it's correlating with. So this is a whole bunch of students at UConn doing this work. So stay tuned. I know she started to present some of these results of the shell analysis, and it's providing some interesting results of what's going on between the Mid-Atlantic and Georges Banks in shell. In conclusion, what we've seen with this project is that most likely sea scallops will experience conditions that will cause growth to slow down by the year 2050. And the combination of warming in a way may cause these benthic habitats to compress and potentially shift them into deeper waters and further north. And as I said, the preliminary model results suggest that Georgia banks will take longer to reach harvest size under warming and acidification. So right now we're doing workshops to engage with the fishing community, to make them aware of the work we're doing, to identify some key concerns and provide educational opportunities, and also work with them to try to figure out some of the ways we can help with acidification as we go forward, so that industry can continue to be a $500 million industry for the United States. Next year will be our last rounds of workshops. We plan to probably be in Virginia, New Jersey, and Massachusetts again. So if you know any industry that wants to come, make sure to let us know. And if you know anyone in the industry that would like to talk to us, please let us know. And that's it. And after me, Jim's coming up to talk to understand the fishing capacity and community ability to adapt. So I will leave it on it at that.
All right, good afternoon. We're going to stay on the theme of shellfish. So this work is also coming from an OAP-funded regional vulnerability assessment. We call it our STAR project, which stands for Shellfish Threshold Aquaculture and Resilience. And in addition to my co-authors, I want to acknowledge the other collaborators in this project, as well as our stakeholder advisory committee. And some of this work was building off of another NOAA-funded project on ocean acidification thresholds. So you'll see a little bit of that work in as well. This project focuses on the Chesapeake Bay region, and in our region, our aquaculture is dominated by bivalve shellfish, which are animals that produce shells made out of calcium carbonate, so they're in theory vulnerable to ocean acidification. And in previous work that has looked at exposure to ocean acidification, I'm really glad that Jim mentioned exposure and sensitivity and adaptive capacity. Exposure, it's essentially has been historically limited by the data that we have, which haven't been as great in Chesapeake Bay and our coastal areas. So when we think about ocean acidification, we know that this long-term change in ocean chemistry is driven by atmospheric CO2, and that is causing a decrease in pH and a decrease in the saturation state of calcium carbonate, and here I'll be referring to the calcite saturation state, which makes it harder for animals like bivalve shellfish to create their skeletons and their shells in this case, and to maintain their physiology. The situation is much more complicated in a coastal environment, including in estuaries, where we have other drivers of changes in carbonate chemistry. And these drivers have been at play for a long time as well, and they create this very dynamic temporal and spatial landscape of carbonate chemistry. This is the natural variability in the systems that we're studying, and it's also a natural experiment that we can leverage to understand how shellfish and the industries that rely on them respond to different carbonate chemistries areas. So here, for example, in the map, you're seeing a projection of bottom pH throughout Chesapeake Bay, the colors representing different pH levels. You can see that pH varies spatially throughout our region, and a snapshot from a time series in 2019 to show how dynamic pH can be temporally. So in this project, we wanted to understand how this natural variability could be connected to aquaculture production in our area. And specifically for this talk, we asked, based on omega calcite variability, how does exposure to acidification conditions vary among aquaculture lease areas, and what is the relative effect of exposure on aquaculture production? And this work would be possible without modeling efforts and collaborations between the modelers and social scientists, like in the project you heard about with scallops. So to dig into this first question, how does exposure vary among aquaculture lease areas? We started by leveraging some long-term time series in our area collected at continuous monitoring stations by the Chesapeake Bay National Estuary Research Reserve Virginia program. And those monitoring stations are in the numbered circles you can see. I've zoomed into part of Chesapeake Bay, Virginia, where the Rappahannock and New York River tributaries are. And these time series include pH among other variables at 15-minute frequencies up to 18 years long, and range in salinity from 0 to 25, which is inclusive of the salinity in our area. You can see that in the color projection on the map there. And what you can also see on this map, are these black polygons, which are the individual aquaculture leases for eastern oyster aquaculture in these same regions. So these data are being collected nearby, relatively nearby, where oysters are being raised for human consumption. These time series, though, only measure pH, and so in order to do this work, we estimated omega calcite for all these time series, and I can go into more details how we do that another time. Because I want to focus on our calculation of exposure to acidification. And here we're thinking about exposure as this combination of severity and duration, where it's meaningful for an oyster how far omega is below this generic threshold of 1, which is a thermodynamic threshold for calcification, and how much time water conditions spend below the threshold. So exposure is this combination of those two things. And in recent work for our OA Thresholds project, we did verify that for eastern oysters, this threshold of 1 is relevant, where you can see in for omega calcite values above 1, we see these positive rates of net calcification, and there's this threshold as omega calcite decreases, where we predominantly see rates of net dissolution or negative rates of calcification. So we calculate exposure over a season at each of these stations where we have time series data. So exposure is what you are seeing on the y-axis, and means annual salinity of the stations is what you're seeing on the x-axis. So you can think of this as distance up the tributary as well. And so a couple patterns pop out to you immediately. First, we see these areas in high salinity regions where there are conditions that are conducive for calcification. We're not seeing this positive exposure to acidification conditions. And then as you go up the tributary, where mean salinity is lower, we are seeing this positive exposure where there are conditions that are harmful for calcification. And we can identify, at what salinity this exposure starts to emerge. One of the other things that you notice are these colored lines, which represent the different seasons. And spring pops out as a season where at any given salinity, there's higher exposure to acidification conditions than in other seasons. So for an oyster farm, which is located in a particular place, it's fixed in place, these seasonal changes may drive a lot of the acidification exposure for their farm in a given year. So what does this look like when we compare to a different tributary where there's also aquaculture production? So on the left is that same figure we were seeing for the York, and now I've added on the right-hand side one for the Rappahannock estuary. We see these same general patterns. We see exposure increase as salinity decreases. We see springtime be a season with relatively more acidification exposure. But one thing we can do to compare these two is to look at a given salinity and see if exposure is the same. So let's do the salinity of 10, and we can see that in the York estuary, we have an exposure of around 65 or 60 or so, whereas in the Rappahannock estuary, at the same salinity, our exposure is maybe 20. So measuring salinity, for example, as a proxy for exposure won't work if you're looking across these different tributaries. So the tributary definitely matters. why that exposure is different is something that we're looking into. One thing this means that exposure is different between these tributaries. We're already seeing relatively more exposure in New York than in the Rappahannock. When we add additional CO2 to the equation through global ocean acidification, it might mean that impacts of ocean acidification may emerge earlier in time in the New York River than in the Rappahannock. So to wrap up this section of the talk, we found that exposure to acidification conditions likely varies among these areas. We saw higher exposure associated with lower salinity locations, springtime as a season, and the York River. And so can we connect these differences in exposure to differences in aquaculture production? Could we see evidence of this variability in water condition playing out in harvest on these different leases? We were able to get these data from the Virginia Marine Resources Commission, which is blinded, but we can share the general results coming out. So for this part of the project, we are looking at exposure in a different way. And this is by looking at it in terms of calcification rate instead of measuring exposure by what the water chemistry is doing. And this allows us to take into account temperature as another driver that affects calcification rate. And in this case, we're using a threshold not of omega calcite equals 0, but when net calcification equals 0. So when the animal is transitioning from a state of shell formation to shell loss. And you can see how net calcification, our function for net calcification, varies across a two-dimensional landscape of temperature and omega up in that plot. But using this threshold, we can calculate exposure using calcification rate, again combining severity, so how far net calcification is below 0, and how much time net calcification is spending below 0. So using this approach, we can calculate exposure over a season like we did for omega calcite at each lease using model output from our group, and I can fill you in more on the details later if you'd like. So we have biogeochemical output from 1985 to 2020, and we can feed that into our oyster model to produce these net calcification rate data. So here's the same map you've seen before, but now I have plotted exposure using this net calcification approach, using winter, the mean winter across the years of the model output. And it might be, I can make out the purple. You can see that there's the purple regions are where we're seeing shell being added, and we see these hot spots of shell loss in orange around Station 10 in the York River, around Station 4 in the Rappahannock River, and up in the Potomac. We can compare that, for example, to spring, where we see much more intense colors across the board, more intense purples with shell formation, more intense oranges with shell loss, and those oranges are occurring in different places in the different seasons. When we pull out and look at the data as a whole, with R for Rappahannock and Y for York, and these are lease level data across all of the seasons, we can look at how the exposure compares between the two tributaries and see that it's similar. We can also look at production in pounds between the two tributaries and see that it's similar. But we see this really large variability in exposure and production among the individual pieces. So we could look into that in terms of variability across the seasons. Again, this is aggregated for all years of the time series by tributary. And we see a seasonal pattern emerge that is complementary to what we saw with our time series analysis, where we are seeing this negative exposure, and this time negative means net dissolution in springtime, where in both tributaries we are seeing from the calcification side evidence of exposure to acidification conditions. When we look at production though, we don't see this seasonal pattern, and of course there are a lot of other things affecting production besides what the water chemistry is doing, like human decision making, which the social scientist colleagues of our team are looking at. But one thing that we do see is we do see a few patterns where production in New York is greater than in the Rappahannock in winter and spring, but it's also more variable. And so this variability is something we're looking into. But this is just to show you a snapshot of the data for exploratory purposes. What we really want to do is link exposure to production. There's a lot of variability. What can explain some of this variability? Can exposure explain some of the variability in production, for example? So we did a random forest regression approach where our predictors were net calcification exposure and scope of growth exposure at the least level, and our predicand of seasonal pounds of production at the least level. And from this approach, we get 3 output metrics: an R coefficient, p-value, and an array of relative importance values for each predictor. And from that, we can narrow in on the specific effect of calcification exposure by multiplying that R coefficient by the relative importance of calcification exposure generated by this approach. So this will be an adjusted R coefficient that I'm going to show you on the y-axis on the plots coming up. So here's our adjusted R coefficient on the y. And again, data are aggregated by tributary on the x. And one thing that we've been able to detect is that this exposure, in terms of effects of water conditions on calcification rate, explains about 30 to 40% of the variation in production on average. And that this sensitivity of exposure, or sensitivity of production to exposure, is similar for each tributary. Again, we're seeing this really large variability in that sensitivity among the different lease areas. So there may be some leases where production is really closely tied to what the carbonate chemistry is doing, and there may be some leases where production is relatively independent of what the carbonate chemistry is doing. And that's really interesting to think about in terms of an adaptive capacity perspective, because those farmers might want to make different decisions. So can we, again, explain some of this variability by season? It's Lindy. When we plot this R coefficient, break it out among the seasons, we don't detect a seasonal pattern in the relationship between exposure and production. Although we do see some differences between the tributaries in certain seasons. So for example, lease areas in the York River are more sensitive to exposure in the spring and the summer than leases in the Rappahannock River in those seasons. So what can we conclude about this relative effect of exposure on aquaculture production? We found this correlation of 0.3 to 0.4 on average. We found that effects of water conditions on net calcification are equally important in driving exposure across the 4 seasons. There wasn't a particular season where water conditions were more important than others, but we did see some season-specific differences among the tributaries. One thing, though, that where we want to take this work next is that for the work I showed you today, we didn't incorporate any time lag between when we're looking at the production, the time point when we're looking at the production data with respect to the time point when we're looking at the water conditions. But we know, for example, that a big acidification event today may not affect harvest on a lease today in terms of how the shellfish are growing. So we want to explore relevant time lags to incorporate into this analysis. So, for example, effects on production may not emerge within the states. So taken together, locations with greater exposure to undersaturated conditions are generally more likely to experience declines in production. We detected that relationship, but that relationship may not hold true for all leases. We saw huge variability in those R coefficients, as we're seeing particular area. And this could be due to other factors that we haven't incorporated in our analysis, like multiple stressors, like timing of harvest, like the potential for aquaculture producers to notice what's going on and change how they're operating their farm. But we still feel that this study provides an important present-day baseline for potential impacts of future coastal acidification on aquaculture of eastern oysters in our area, which will be useful context for anticipating future ones. That's it for
Okay, so my name is Cassie Gervis. I'm faculty member at St. Mary's College of Maryland in the new Department of Marine Science. I'm also a seagrass SAB ecologist, and I was asked to just kind of give an overview of what we know about interactions between acidification and shifting gears, the plant world, so SAB and seagrass. So that's what I'm going to do, is kind of highlight some recent research on this topic. And just to orient you, I'll be talking about kind of seagrass and SAB as like a collective whole. So a common seagrass species in the mid-Atlantic is Zostra marina, or eelgrass. And then we call the very similar plants that live along estuaries, salinity gradients, verged aquatic vegetation, or SAB. And there's a whole bunch of different species that would be considered SAB. So I'll kind of talk about them interchangeably. They're functionally and structurally pretty similar, although there are differences too. And I'll be focusing on these three kind of overarching questions. So how did increasing CO2 and temperature affect SAB? So what are the effects of acidification on the plants themselves? And then how did SAB in turn affect carbonate chemistry? And then can that effect help to mitigate some of the negative effects of acidification on other organisms like these calcifying bivalves? Start with that one. And when we think about how stressors in the environment might affect SAB, it's useful to think about it in terms of the plant carbon balance. So as plants photosynthesize, they're pulling CO2 out of the water and converting that into particulate organic carbon in their tissue. And they're also respiring as well. So they're taking some of that organic carbon and then converting it back into CO2 and releasing it into the environment. And so if they are photosynthesizing more than they're respiring, they can maintain a positive carbon balance. And they're doing okay, they can grow. If they're respiring more than they are photosynthesizing, they go into the state of negative carbon balance and become really stressed and they can die off. And so thinking about whether the plant is in a state of positive or negative carbon balance relative to different things going on in the environment can help us kind of understand what's affecting SAB. And so one of the things that I guess the most relevant thing here is if you're increasing the CO2 in the environment, then that could, in theory, possibly help them photosynthesize even more and maintain a positive state of carbon balance. And this study, this is by Dick Zimmerman, where they were working with Zostra marina, so that's eelgrass in the lower Chesapeake Bay. It's a mesocosm experimental study where they had a whole bunch of different tanks. They were growing eelgrass and they bubbled some of them with CO2. And you can even see just from the pictures, the top picture is the highest CO2 treatment and the lowest picture is the lowest CO2 treatment. And so adding CO2 clearly made the plants accumulate more biomass. And they also have this correlation plot where as you increase pH, which is less CO2, there's less biomass accumulating. Lower pH means higher CO2 exposure, so you have more plants. There's this positive response. And this is another study by Dick Zimmerman and colleagues where this is a modeling study here, and they're showing that growth stimulation from the increased CO2 concentrations can even offset the negative effects of increasing temperature. So, it's not just CO2 that's increasing, the temperature is also increasing. And eelgrass in particular is really sensitive to high temperatures. And that's because increasing temperature increases the rates of respiration. So the plant goes into negative carbon balance,
if it's not photosynthesizing enough and that it's respiring a lot more, we get a lot of plant loss that way. And so this is a model run where they're showing the distribution of SAB in the lower Chesapeake Bay with a 5-degree increase in temperature from ambient. And so you can see there's some SAB loss there. The shades of green represent plant density. So we have less production there and some die-off. But when they model the same system with both elevated temperature and CO2, we don't see that die-off. And so that's because the extra CO2 stimulates the plant growth enough so that it can withstand those high temperatures. But that's not necessarily always the case. So eelgrass, in particular, also just prefers to use CO2. But a lot of other SAB species don't need to use CO2. They can use bicarbonate, which is another species of inorganic carbon. And bicarbonate is much more abundant in aquatic systems than CO2 is. And so if you have species that are using bicarbonate, if you add CO2 to the system, they're not going to care because they're not carbon limited anyway. And then also if there's other factors in the environment that could be limiting photosynthesis like light or not enough nutrients to support plant growth, you might also not expect to see a response to that increased CO2. And so this is just one example of a study that showed pretty much no effect to CO2 treatment. So this is a mesocosm study with Helidule and Rupia. These are two seagrass SAB species. I think this was from the Gulf of Mexico. And so in this, the 3 figures are just different measures of plant production, and the blue is ambient CO2 and the red is elevated CO2. So there's some kind of visual qualitative differences, but none of those were statistically significant. And they are kind of hypothesizing in this study that the reason is because those plants are really good at using bicarbonate, so adding CO2 just didn't have any stimulating effect on them. So yeah, basically the effect of increasing CO2 on seagrasses really just kind of depends on what species we're looking at. So how does SAB affect carbonate chemistry? So one thing is that plants are, seagrass systems in particular, are very productive. This is a synthesis of global estimates of seagrass net ecosystem production from all around the world. And because most of these values are above 0, it means they're net autotrophic. So they are fixing more organic carbon than they are respiring. So these systems can pull a lot of CO2 out of the water. And so in theory, as the SAB draws down the CO2, we get a shift in the carbonate system, right? So whereas acidification is adding CO2 to the water and hydrogen ions, so we get more acidic water, and the system shifts away from carbonate saturation. When you have seagrasses putting a lot of CO2 into the water, it's the opposite, right? So pH goes up, and you have this shift towards higher calcium carbonate saturation. And yeah, that's basically what I just said, but with looking at it in a different way. And so sometimes in seagrass systems, they can increase the ambient CO2. So we see like definite signatures in pH in the water column. So they can draw down CO2 so much that you see that signature in the water column, but then also there's like these microzones, right, like along the surface of the leaves where the pH gets even higher. And as the pH gets higher, that's kind of showing that also the calcium carbonate saturation state is increasing as well. And you can get chemical precipitation of calcium carbonate on the surface of leaves and also like within the cellular structures of seagrass leaves. And that's what these are scanning electron microscope images of some seagrass from the Bahamas on this side, and on the right is from Chesapeake Bay. That's wild celery from Susquehanna Flats in the Upper Bay, both showing these precipitate on the surface of the leaves. So that's I think that's super interesting. And also, that precipitation process has an effect on the carbonate system, right? So it releases some CO2 back into the water, shifts the carbonate system around. There's also a lot of calcifying organisms that encrust the leaves of SAB. So there's a lot of additional calcification going on within seagrass systems that wouldn't be happening if the seagrass wasn't there. It's kind of home for these organisms. And seagrass and SAB can alter sediment biogeochemistry in ways that also affect pH and CO2 and alkalinity, all these parameters that are relevant. So the roots of SAB can actually leak oxygen into the surrounding sediment, so that affects redox chemistry. They also add a lot of organic matter to sediments, which affects redox chemistry. And it just gets really complicated, and you add the calcification and dissolution processes that are also amplified in seagrass beds. You have kind of a potentially big and complex kind of interlinked effect on carbon chemistry. And there's a lot of ongoing research to kind of get into, dig into some of those processes. And I'm part of just one project that is looking into that, where we're doing these in situ incubations in seagrass beds at a few different sites around the Chesapeake Bay to look at the fluxes of alkalinity and dissolved inorganic carbon. Also, Emily's part of that project. So that, we kind of just started that this year. All right, and then the last question is, we know that all these processes do happen, they can happen, but is it enough to actually mitigate the negative effects of acidification on other organisms? So this is a study that showed that there does seem to be a positive effect. So they, another mesocosm experiment where they were growing oysters, Crassostria gigas, with eelgrass, Zostera marina, and they were exposing it to, so they were exposing the oysters to different densities of seagrass and measuring pH and carbon chemistry and a bunch of other parameters. And they saw this positive relationship between shell growth and seagrass density. So shell growth on the y-axis, seagrass density on the x-axis. And they attributed that to the increase in pH and increase in the calcium carbonate saturation state that they also measured in those same treatments. But we don't always, you can't always expect to see that effect. And there's a lot of other studies out there that in, either in situ or experimentally showed that there's not an effect. Well, especially the in situ, so you can't always expect to see what's happening in these experiments in the field, because the field is a lot more dynamic. And so this is a modeling study that was, I think, did a really good job of showing how the relationship between hydrodynamics and plant production really affects how much of the pH signature you're going to see in the actual environment. So they have residence time on the x-axis. So residence time is like the amount of time a parcel of water will stay within the seagrass bed. So as residence time increases, the effect on pH, that's where delta pH isolines are, also increases. So you have bigger increases in pH as residence time increases. And that's because, conceptually it just makes sense, because you have water, it's sitting there longer, there's more time for this seagrass to do their thing, to kind of draw down the CO2, increase pH, and there's less mixing with the ambient water. And then also as you increase seagrass production, that's on the y-axis, you also see these increases in the effect on pH. So these really productive systems that are big, dense SAB beds, where there's a lot less mixing with the surrounding environment, you're more likely to see that effect on pH. Whereas if it's a small or patchy SAB bed, it doesn't even matter how productive it is, you're probably not going to see much of the signature. And so this is that same modeling study where they parameterized the model for a system in California, and they showed that these lines here are lower velocity, so residence time is related to water velocity. And so the lower velocity model runs, there's really no effect on pH. And the effect on their these are the higher velocity runs. The lower velocity, so residence time is going to be longer. There was an effect, but it was still not really a big effect. And because they think this system is like a pretty small patchy bed. So there's really not really much of a chance for that, even though the plants are productive, for them to kind of work on the water. Okay, so there's this is another study kind of projecting into the future what we might expect to see in terms of seagrass effects on pH and exposures. And so they are kind of focusing on this idea that the buffering capacity of seawater is also going to be decreasing as CO2 concentrations increase. And this is really particularly important in dynamic coastal systems where you have these really huge diurnal swings, especially in seagrass systems that are really productive. So they're increasing pH at night during the day, but then they're also decreasing pH at night because the systems are respiring. And so they do these model runs from pre-industrial times, this is 2015, and then projecting out into 2100, and you're seeing an overall decrease in pH. So the median is decreasing, but also just the magnitude of those pH swings on a daily basis is increasing as well. And then they took that information and then looked at exposures to jugal, or I forget if it's juvenile or whatever, larval bivalves. And so they have a model with seagrasses, a model without seagrasses. The dashed lines are favorable conditions, the solid lines are unfavorable conditions. So you look at this solid line for the seagrass model, the larval bivalves are going to be experiencing more unfavorable conditions than compared to the model without seagrass, at least until this kind of threshold here. But they're also experiencing a lot more favorable conditions. And so they're kind of highlighting the importance of research like Emily's doing. It's not just the shift in mean conditions that's relevant, especially in these really dynamic coastal environments. The shift in the extremes is also really changing and something we need to understand better. And then just really quickly, I'll kind of, this is another study that this is looking at system scale effects now. And they were looking at the carbonate chemistry along the axis of the whole Chesapeake Bay, and they found this kind of interesting behavior. I forgot to put, this is Jinzan Su. He's a grad student at Wei Jung Kai working at the University of Delaware. They saw that if this is the kind of conservative mixing line along the this Chesapeake Bay salinity gradient, they're seeing drawdown of all these parameters of the carbonate system in the upper bay and then release of it back into the water in the mid bay. And so what they are hypothesizing happened is in the upper bay is this huge, huge, huge SAB bed. I've done a lot of work up here. This is what it looks like on the ground. It's like, I think, 5 kilometers across. And part of my graduate research showed that there's quite a long residence time inside that SAB bed. And so and also pH gets really high. During the summer, it can even get higher than 10. So the SAB are really cranking the pH up. And they measured calcium carbonate in the plant biomass and also did these scan electron microscope images and found it on the surface of the leaves. And what they're hypothesizing is that some of that calcium carbonate is being advected down into the mid-bay where you have hypoxia, and so you have acidification due to high rates of respiration in the bottom waters. And so they think that some of this calcium carbonate is dissolving down there and then buffering some of that acidification. So that's kind of a cool, unexpected sort of cross-scale effect of seagrasses on acidification. Okay, so just to kind of recap, so just how does CO2 temperature affect SABs? So sometimes it grows better, sometimes it doesn't. It just kind of depends on what species you have. SAB can increase pH and calcium carbonate saturation, but it's kind of complicated, right? There's a lot of interacting processes going on in SAB beds, and whether or not it can help mitigate acidification kind of in large part depends on how productive the system and how well mixed it is with the surrounding environment. There's a lot of other complicated things I kind of glossed over, but I think those are kind of the main components that are important to be thinking about. So that's it.
So if you want to come over here for a minute, yeah. We have about, we're running a little bit behind them, right? But we'll do about 5 minutes for questions. So people have questions, go ahead and raise your hand, and I'll run around with this microphone, and we'll have the group of speakers go ahead and answer them for you. So, Erica?
Just because I know this work exists, I wanted to ask Emily to maybe chime in a little bit on some of the really, you mentioned in your presentation some of the thresholds work, but I wonder if following that last presentation, you want to maybe expand on that with, anything you're thinking about how OA and seagrass beds might be able to mitigate, and if you have any further clarification on that.
Yeah, sure. Thanks, Erica. So our OA threshold project was focused on looking at potential interactions between eastern oysters, so Crustacea virginica, and SAB, in particular eelgrass, from a modeling perspective to see if there were scenarios under which there could be positive interactions between these two species, in terms of the way that both of them manipulate the carbonate chemistry of the environment for the other. And Cassie really nicely summarized that in the publications that have come out of it. Our publications are still forthcoming. But we did find scenarios, we explored different densities of oysters, different densities of seagrass. Our model environment was not a spatial environment like what I was showing here in this other project. So that would kind of be a next step of that work, is to be able to put that in a more spatial setting to look at, okay, well, as hydrodynamics are changing over a transect of a bed from the edge to the center and out to the other edge, or how contiguous the bed is, how that would influence what the growing environment might be like for a calcifying organism. But we did find scenarios where there were densities of oysters that benefited the growth of the seagrass by improving water clarity, for example, and that there were densities of seagrass that benefited oyster growth by enhancing the carbonate chemistry in the ways that Cassie described. So this was purely in a modeling space, and so another future direction of this work would be to try to do in situ experiments with planting of oysters in seagrass beds and things like that to see how these would play out in a natural setting where there are other environmental parameters at play. But I think, our region and our group of collaborators here is a wonderful group for thinking about moving that forward, because we have a lot of SAB in our coastal areas and a lot of shellfish production, a lot of shellfish restoration, and we need all the help that we can give these systems in the future. So, thanks.
Thanks, Emily. Do we have any more questions? Okay.
Thank you. I'm Mike McMahon from MD at the Chesapeake Bay Restoration Section. We work most of nitrogen, phosphorus, and total suspended solid reduction. Obviously, suspended solids and sediment can reduce down light to the seagrass beds. Can it affect calcium, interact with calcium and pH? Is it a factor?
Well, so I'm not a chemist.
Yeah.
I can answer that from the ecological side of things. So if you have less light, then the plants are going to be less productive, and so they're not going to be photosynthesizing as much and drawing as much CO2 down out of the water. So they would affect the carbonate system, and that would be like this sort of indirect effect where those parameters are affecting photosynthesis, which in turn would affect the CO2 concentration. Whether there's a direct interaction between nutrients and calcification, calcium, I'm not sure about that.
Thank you. I think we have time for one more question. you guys very much. We'll go ahead and shift over to the next session. Thank you.
Okay, our next session is on new initiatives in outreach and community engagement. So our first speaker is going to be Jennifer Freeland from GCAN. Then we'll have Emily Silva from MACAN, so there we go, more cans. And then finally, for the third talk of that session, is Jessie Turner from the OA Alliance. There we go. Hi, everybody. My name is Jennifer Freeland. I am the program coordinator for the Gulf of Mexico Coastal Ocean Observing System. I'm also the coordinator for GCAN, the Gulf's Acidification Network. I am a marine biologist, and my background is on red tide, harmful algal blooms, and ocean acidification. And I just want to thank MACAN for having GCAN come and present today. We have a very close relationship actually with SOCAN, and the project I'm going to be talking to you about today is a collaborative project from GCAN and SOCAN. And it really was led by Dr. Janet with MACAN. So the project that we worked on, we decided, just to give you a little bit of background, GCAN is one of the youngest cans. And we are looking to really kind of identify what our community is feeling, or their knowledge base about ocean acidification. And thankfully, SOCAN is also, I mean, they are definitely more, have more of a listing of participants than GCAN has. We have a little ways to go. But, you know, we felt that this project in particular would be really beneficial for both of us to learn about the community. So let me just, and I'm going to try to spell out all the acronyms, so bear with me, okay? So this project is called the Coastal Acidification Network Stakeholder Feedback Project, which probably should be changed to the Community Feedback Project, okay? So bear with us on that as well. So the motivation for this project is to support the NOAA's IWGOA Communities Vulnerabilities Assessment, which was just released today, so that's great. The Monitoring Priorities Report. And it was also based on a little bit on what I just mentioned, which is to begin to understand what the general understanding of OA is within our region. So then we can develop strategic materials and messaging for the community. So that was our main focus for this. The other type of, you know, our project goal, more specifically, the goal is to understand what the invested community knows about acidification, how they are impacted, and what they believe our monitoring priorities should be and where gaps are currently. So I have a couple of benefits on a broader scope. These benefits, you know, consist of a survey with results that will help support and build upon federal reports, and also from other OA-specific surveys in the works, both regionally and nationally. For example, the OAP National OA Survey, which is currently in development. So we're really looking forward to helping with that, and just kind of start to develop and build upon a lot of questions that we have. I can tell you for GCAN, we have a lot of expertise within our membership and panels, but we don't have a lot of stakeholder engagement. So that's something that we really need to benefit from. So the survey, so we have this survey. We were looking to find a social scientist, and that proved very difficult in our region, especially with budgeting. So we looked onto our national estuary programs, and we developed our surveys using the methodology from the CHNEP, which is the Coastal and Heartland National Estuary Programs Comprehensive Conservation Management Plan, the CCMP, the Climate Change Vulnerability Assessment, and also input from the CHNEP staff and committees. So this is where we based our survey from, and they were a huge help. Jennifer Heckler from the CHNEP was a very important asset to this project. So in the Gulf of Mexico, we know a few things, but there's clearly a gap. So you could see, we have based on this 2019 jobs and economic revenue from the recreational and commercial fishing industry in the Gulf of Mexico, there is quite a list of economically important fish. There is a shorter list, but a growing list of studied species. What we do have a clear gap on is wild species impacted from these activities. So, you know, overall, we know a lot about human activity, but we don't know a lot about OA affecting the wildlife. So more work needs to be done on that. You can also see here, we have quite a lot of jobs that are dependent on this industry, on commercial and recreational fishing. Employment income is very dependent, and so is our gross sales for this industry. So finding out more about the general knowledge of OA is super important when you're considering strategies in research. And then we have the Southeast. It's from the same 2019 U.S. jobs and economic revenue. There's even less studied species, surprisingly enough. We thought the Gulf of Mexico was pretty late in the game for a lot of things, but you can see also wild species impacted, also a huge gap. So we really need to know where we want to strategize areas of research. So we feel the best way to do this is to just start asking questions and getting feedback. Also, that's important for the Southeast is their jobs. For this very same, regional, recreational and commercial fishing, employment income is quite substantial, and so is the gross sales. So the survey we put together, we did it during COVID, as COVID was improving. So we were able to circulate this two ways. So we did it through Mentimeter. You're probably pretty, or maybe you're not aware of Mentimeter, but it has a lot of qualities that you could do online. So we set up a Mentimeter and Google form, both of which you could do online. You could do it in person, or Mentimeter in person. But what we split it up, we did virtual presentations, and we were able to actually get out and personally be involved with some of the meetings that were held. We looked through the MEPs, and we presented this survey between their citizen advisory committees, and we felt that would be the best way to get a hold of our primary stakeholders and users. We also presented through their technical advisories. So this was NEPs around the Gulf of Mexico and
and we utilized their meetings through, and they were kind enough to give us time to present. So in total, we had, through these surveys, we had 177 participants and results. So we felt that that was a pretty good number. We did circulate this we had the survey attainable through the MACAN website, not MACAN, SOCAN, sorry, SOCAN website and GCAN website. We circulated through our e-newsletters, and the Mentimeters we kept pretty much in person. So we were able to, while we were presenting, they were able to, we could tutor them through the Mentimeter. So that was very interesting as well. So moving on, as we were developing this survey, we wanted to break it up into some general categories so that we can identify what we wanted to ask and the best questions moving forward, depending on the region that we were presenting to. But we had to keep it so that all the questions were cohesive, everything stayed the same, so it was very challenging to come up with these questions, but we did it. So some of the survey categories: general knowledge of OA and the ecological anthropogenic processes. Then we had a level of concern about OA impacts, direct or indirect. We had a category of recreational activities and resources in marine environments. And then we had our participants ranking OA priorities. So I'm going to walk you through some of these questions. We worked on trying to make it as short and sweet as possible, but lessons learned as we will go through. The more efficient and streamlined you are, the more participation and attention you have from the participants. So this is really kind of the first and foremost survey that the CANs have led as far as general OA stakeholder feedback. So the general knowledge of OA, do you have any knowledge on this topic prior to today? And our overall consensus was some. So it's really sort of consistent. We know about OA, but we don't know a lot of the details, sort of, so to speak. Was the general feedback of what I had gotten in person. So how would you describe your main source of information about acidification? Our top selection picked was school and academia. So, and this again was through both our technical advisory committees at the NEP and our citizen advisory committees. So, which of the following processes are you familiar with? Number one pick was nutrient contamination and eutrophication. We don't know if this could be because there is a very known observation and understanding of red tide in the area, which is very similar as far as their knowledge base. We really, especially in the Gulf of Mexico, work to educate the communities on red tide, and so there's a lot of knowledge of nutrient and contamination, eutrophication in that region. So whether or not that response was because of that education, we don't know. So it's just kind of that catalyst that we can now find out information and look deeper into. 4, which of the following processes do you think could lead to or worsen acidification? Number 1 choice was nutrient contamination and eutrophication. So again, there is a building of knowledge in the region of effects such as red tide, harmful algal blooms, but again, we don't see a lot of ocean acidification outreach in that area for many reasons. So that was very interesting for us to see. Category 2, level of concern about OA impacts, direct or indirect. Are you concerned about the effect of acidification will have on the environment? So yes, the majority said absolutely. So there is some concern. Number 6, if you are concerned about the effects of acidification on the environment, please rank the ecosystems on concern to you, of concern to you. So estuaries, bays, and lagoons, number 1 choice. And then it goes on, are you aware of the potential effects of coastal or ocean acidification on marine animals? Yes, overall consensus, yes. So they're concerned about OA, they know something about OA. Of the marine animals that may be affected, which are you most concerned about? And the number 1 was corals. So that in itself was very educational for us, being that overall consensus between both citizens and the expert committee. And then, are you concerned that acidification may affect your employment in the future? And it was no. And it was interesting because most of them, of the participants, said if there is ocean acidification present, that will keep my job going. And so I really don't have to be concerned with it. So that was an interesting spin on things for us. So the following locations could be affected by acidification. Rank your concern about the potential effects of acidification. So that was shallow coral reef ecosystems, and then moving on to category 3, recreational activities and resources in marine environments. Do you fish recreationally or commercially for finfish or shellfish? That was 50-50. 100% were recreationally, but there were no commercial fisheries people in the poll. So another interesting flag for us that showed a gap was that there were no commercial fishermen a part of any of the NEPs that we had surveyed, which is a gap. So that was very telling for us. Moving on to 10, if yes, please specify which zone best describes your fishing grounds. So inshore, bays, and estuaries. And then we have 11, if these ecosystems are affected by acidification, which in your opinion is most important to focus on. And in their opinion, it was nearshore and coastal waters. And then what describes the top 3 types of seafood you fish for or consume at home? And the overall was shrimp. So again, that was another really interesting find for us within that community. So again, when we are developing educational materials, outreach materials, this gives us some insight on how we can pinpoint our focuses. And what recreational marine activities do you take part in? Scuba, which was very interesting to us. Out of all the other, going to the beach, kayaking, paddleboarding, sailing. So we have some diehard scuba people in the Gulf. So. And then our last category 4, ranking OA priorities. Rank aspects of acidification research that you think should be prioritized. And the number 1 was impacts to habitats. Then I skipped 17 because that goes farther down into this, yes, that's at a different slide, sorry about that. So number 18, rank the following educational opportunities that we should target for acidification, and the overall response was policymakers. So that was another very telling aspect for us. It just so happens that GCAN had been looking to create a policy advisory group, a working subgroup that can focus more on policy. So that was really kind of a win for us because we know that we needed that. So number 19, what type of communication would be helpful for conveying information about acidification? And social media was number 1. So we had perceived that being the case, and we know that we do social media, Instagram and Twitter, but we can definitely improve on that for sure. So that was another highlight for us that we knew we needed to improve before we got the results. So that'll help us focus on our strategic agenda moving forward. So, and then we had quite a list of, we had an opportunity for the participants to add areas where we left out. We felt this was important for them to voice their opinion and help us on, surveys moving forward and ways that we can help with other surveys on a higher level. So I thought interesting, better understanding the relative role of global climate, chance and atmospheric CO2 levels versus nutrient inputs to acidification from air pollution, human health, and plant impacts. So that was an interesting answer that I felt like there's this, again, they know that acidification is there. The details are blurry. They know they should be concerned about it, but not quite sure how to proceed forward in ways that they can help. And then I just kind of went through and combined everything so you can kind of see, like, there's familiarity with OA and our overall answer goal. There's concern. There is awareness. In this particular project, the overall concern is for corals. And it really kind of gives you this overall picture of where we stand as far as the Gulf of Mexico. And we really, we're going to take this information, and we're still processing through the info, but to just give you a nice little highlight, on the Mentimeter, we had an overwhelming amount of Floridians. On the Google form, it was a nice mixture, but again, overwhelming Floridians. 177 responses total. We thought we did pretty good there. And again, this picture will help us really sort of correlate what we want to do in the future. We have all this ability to be able to do outreach, but again, because we're young hands, we're just trying to get a feel of, what the general consensus is. We can really take this and strategize moving forward with education, materials, that so to speak. So, lessons learned. There were a couple of good ones. We had some technical issues with the Google Forms, and that can get in the way depending if you are a PC or Mac user. We decided better methods, we need better methods for contacting stakeholders, maybe getting in on additional meetings, branching out, and so to speak. But we had a timeline, so this is what our best scenario was in order to really reaching those end users. Interpretation of questions can vary, so we have to be very cognizant of that when we are creating a survey, and the participation group that you could be attracting, and what their overall viewpoint is in general. This could be political, this could be religious, this could be in any sort of way, something that you have to be cognizant of when you're developing these questions. Keep presentations to 15 minutes or less to allow time for all the questions. We had a lot more questions. Didn't we, Janet? I think we started with a lot more, and we had to really trickle it down. So even, and I can recall just being at a couple of those meetings, while we were all going through the Mentimeter survey, I could tell at some points there was some lost interest after a while. So really getting it down to a couple really good questions to fulfill your needs or what you're looking for is probably the most ideal. And then going through the questions afterwards and doing a data analysis, setting up the questions and answers so that they're cohesive and easy for data analyses. Depending on, a ranking system or a ranking questionnaire, that could be difficult in data analysis, depending on if you could set up your questions and answers based on the best type of data analysis you're going to get without having to really kind of break it apart, is also very valuable. But I think, in summary, this was the first attempt for a NOAA OAP CAN survey for end users. Still sorting the data, so there's a lot to be had with that, and hopefully we'll get a nice technical report out of it. Again, we will be reporting this to the NOAA's OAP. We'll be part of the NOAA OAP nationwide survey for decision making on prioritizing, monitoring, and assessment. And then ultimately the report that just was released and headed to Congress. So with that, I think that's it.
Hi, my name is Emily. I'm the Maracus administrator and actually the former MACAN coordinator. Our current coordinator, Austin Pugh, is in the process of moving from Vancouver Island all the way to New Hampshire to join us at Maracus in Portsmouth. So he couldn't make it today. So I'm subbing in for him. As Janet said earlier, a lot of the CANs have similar missions and goals, so I don't really need to explain that to all of you here, who NECAN is. But sometimes they're structured a little bit differently. We have a steering committee and 4 working groups shown here. We have education and outreach, industry, management policy, and science. And they each have their own set of goals and actions that they all work towards. And within these groups and the steering committee, we have representation from state and federal employees, researchers, academics, industry members, NGOs, and other regional partnerships. And also because MACAN straddles in international work, we also have Canadian representation as well. So one of the things one of our working groups has done was create this summary of OA actions in the MACAN region. In 2021, they put this report together. And this was because several states in our region had created commissions or task forces about climate generally or OA specifically. And they had some meetings and they created some reports to show their findings. And so this document was created to summarize the findings of each of those reports in each of the states in our region. Some commonalities, there were several commonalities between all of these reports that had come out. So we highlighted them here. More monitoring, always more monitoring, is needed to understand long-term trends and effects of OA on environments and species. There's interest in learning more about carbon sequestration and marine CDR. Co-stressors and interactions of OA with other variables such as nutrient loading and management, freshwater runoff, hypoxia, habitat restoration. More research wanted on the impacts of OA on commercially important species, and MACAN has actually created a one-pager which sort of addresses this, but it's definitely clear that more research is needed. And then what we're talking about in this session, continued engagement and outreach around OA and education on OA in general. So another commonality was that though these reports cited that we need more monitoring on all of these topics, they don't state what kind of monitoring is needed, where or how often, seasonality, information on various tech and sensors, different methods. Understandably, they lack these finer details. So, but there are several programs in the Northeast that could address these gaps, such as MACAN, Miracus, the Northeast Regional Ocean Council, Sea Grant programs, NEPs, National Estuarine Research Reserves, and others. These programs and current monitoring can be leveraged along with new efforts to fill those info gaps. So MACAN decided to create a regional monitoring plan to attempt to address these questions and more. But to get to the information we needed, we decided to have to hold a workshop. So we were brainstorming all the information we wanted to see, and decided that a 1 or 2 day workshop just wasn't going to be enough to cover all of the different topics we wanted to get more information on. So here's where some of our outreach came in. Typically, MACAN hosts a webinar series every year. And so we decided to hijack the 2023 webinar series for ourselves and host that as sort of an informational gathering activity to put together what we want this workshop to look like and then create a monitoring plan. So overall, the theme this year was monitoring priorities in the region, which is pretty big. So we had each webinar focus on a different topic. And we did advertise this to the wider MACAN community. The steering committee was the primary audience. So we thought about all the various theme areas we wanted to hear about and from whom. And then we adjusted our typical webinar setup, which is usually 60 minutes. We extended to 90 minutes. And then we had more of an informal conversation at the end of each set of speakers, rather than a formal Q&A, which turned out to work really well. You don't have to read all this, but in addition to that list of topics and who we wanted to hear from and from what organizations, we brainstormed some guidelines that we wanted to give the presenters as they were pulling their talking points together. We didn't want to just hear about their research or work in a vacuum, isolated from the bigger picture. We asked them to put their work into a broader context, to provide an overview of the state of the science in their field, along with any recent advances, if it can be augmented by existing monitoring efforts, if new monitoring projects are necessary. And so, as kind of a general overview of the series, it's been a huge success. We've had 12 webinars from March to September, covering 8 theme areas, which I showed on a couple slides back, but they were current assessments, OA and climate, modeling biological impacts, current and new technology, sensors and methods, using new products, indigenous interests, concerns and perspectives, and rapid response. We've heard from 31 different presenters from almost as many different organizations. And we're creating webinar summary papers. And by we, I mean Beth Turner is writing webinar summary papers after each of these webinars, which at first were just going to be a resource for the MACAN steering committee as we're shaping the workshop. But as the webinar series progressed, we decided that we would make these resources available on our website. And so I just wanted to show here and acknowledge all of the people and organizations we've heard from, getting input on all these questions we've asked them. It was a big effort, and it was a diverse group of people. So I just wanted to show how wide that net is that we housed. So this brings me to the workshop. This is coming up this fall. Our overall goals are to identify OA monitoring needs and uses of OA data, explore new ways of monitoring or additions to existing monitoring to help meet those needs, and foster collaboration across the region. So the webinar series was, again, just like an informational gathering activity. The workshop is where we're really going to be able to dive into those details and have in-depth conversations with our community to start shaping the actual monitoring plan. Our hope is that this plan will help state agencies and others to advance those recommendations from those reports, from the OA and Climate Commission reports, and to be used in tandem with not only those reports, but the IWGOA reports. to determine the best way to implement monitoring in the diverse environments along the New England coast. And then, so that's kind of like the big project we've been working on for the past year and a half. But while we were talking about community engagement, I wanted to throw a plug up for some other documents, products that MACAN has helped create with the help of outreach and engagement. So hopefully many of you have seen this before. This is Vocal. This is a set of four kind of one-pagers that we've created. Vocal stands for Visualizing Ocean and Coastal Acidification Locally. These were done in collaboration between the Northeast and the Mid-Atlantic with funding from Sea Grant and NOAA North Atlantic Regional Team. So the first two, this one and this one, provide an overview of some of the drivers of OA in both of these regions. And the reverse side of them, this is the front and then that's the back, just kind of goes into a little more detail about what those drivers are and what they do. And then the other part of Vocal, these two pieces here, one is for industry members, the other is for policy. So again, this kind of just gives an overview of OA in the context of industry and commercially important species. And then the other is more policy related. So it shows, you know, what drivers are in the region, what they do, can we do anything about them, are there any policy actions that can be taken to address them. And then one last piece I wanted to show you was this OA impacts infographic we created, which I mentioned very briefly earlier on. So this came together with help also from NOAA, but also from RARGON, the Regional Association for Research on the Gulf of Maine. And so it kind of just highlights, this is on our website, so you can see it in much better detail, but you can see there's species here, their commercial value, how much we know about them, if there's any studies done on them, the species role in its ecosystem, and then the known effects of OA on its species. Yeah, so that's all I have today on our community outreach. Our workshop is coming up this fall. We're struggling with the dates. It was originally in October, but some of you may know that NOAA is switching over their financial system right now, so that would be there would be trouble for NOAA employees to come to workshops. We're switching it to later, so stay tuned. And this is my information and the current coordinator's information. So. Thanks.
Follow along with the product that I am speaking about. So I'm going to send that to you right now, if that's all right. Does that sound okay? And great. And I've gotten a great job of giving you guys the right before 3 o'clock coffee presentation, so I'll be fast, and this will be a little bit more high level probably than some of this stuff. It's a little bit more from research deep dive. So, all right. Oh, we're not over here, wait. Okay. Great, well, hi, guys. My name is Jessie Turner, and I'm the director of something called the International Alliance to Combat Ocean Acidification. And I will spare you the great background details of what that organization is, but it is a collection of governments, both U.S. states as well as national governments, ports, tribes, First Nations, municipalities even, and a suite of affiliate members, which are what you would imagine, NGOs, science research institutions, intergovernmental collaboratives, working together to better help governments better understand and address impacts of ocean acidification domestically and then through international frameworks. And if you're interested in learning more about the OA Alliance, please come see me. I would love to share more. We have a website that you can navigate through. But this presentation is to give a little bit more context on a really fun comms project in a way that I'm not really used to doing, so it's new for everybody, that
was put forward by a group called the Aquarium Conservation Partnership. They are an affiliate member of the OA Alliance. It is very much what it sounds like, a group of U.S. aquaria coming together to organize themselves, kind of leverage their political voice and various requests to Congress around marine conservation, but also working to leverage their platform as they're reaching out to visitors to U.S. aquaria. And a lot of my time with the Alliance is spent with governments. We're sort of a low capacity but mighty group, and so I really try to engage our governments more directly with a lot of hand-holding around creating these OA action plans, which we'll be hearing about the MACAN variety of OA action plans in a bit. But that means that I've been leaving a lot of affiliate member partners on the table and these great outreach vehicles that we haven't really given them much to do. And so I'll just quickly go through and talk about what this collaboration is. As I mentioned, it is the Aquarium Conservation Partnership coming together with the OA Alliance and then NOAA OAP to think about how we would organize telling stories around ocean acidification that are sort of place-based. And so the idea here is that we have an opportunity to present localized information on unique impacts, responses, and calls to action on ocean acidification taking place across the country, particularly utilizing the cans as these hubs. And so I thought, well, this is great. ACP has this membership of aquaria all over the US. I know that the cans are regionally coordinated and have enough of a deep dive where you can really get into place-based local trends, impacts on keystone species that people are seeing and hearing about and coming to aquarium to learn about. And that we can really think about cross-pollinating those sort of bucket of information that the cans already have. You know, no more researcher work needed. We can package up a lot of that work in storytelling and use the aquaria to be the vessels of that information. And so what that has resulted in are these 6 regional story maps. I'm a very boring, low-tech person, so when I was first thinking about this, I was like, great, we'll do PDFs. We'll do 2 pagers, 1 per region. We'll hand them out to the aquaria. You know, they'll put them up on, I don't know, on poster boards. But luckily, somebody named Edith Marie, who you might know from the Alaska can, came to me and said, I have some time. I'm very interested in comms. Is there anything that you have I could be working on? And I said, oh goodness, yes, let's make these 2 pagers. And she said, no, we're not doing 2 pagers, we're doing regional story maps, and this is what they're going to look like. So the idea here is that each story map has sort of these uniform components, although of course the middle part that's unique to each region obviously reflects the unique vibe of that place. So we have an overview of climate ocean change, including introducing ocean acidification. Like I said, a synopsis of trends and impacts to species and human communities most at risk. A highlight reel of activities, that means a lot of the work that you're hearing about, taking part in locally with stakeholders through the cans, other marine institutes. And then, because I come from a policy background, I was like, oh, and we can talk about all the policies taking shape to address ocean acidification, both at a federal level, maybe state legislative level, and local level. And then the thing that I think is the most unique for me and my experience about this project is moving then into a call to action that would be for an individual visitor to an aquarium. You know, and I think we struggle with that as a community, which is what is our call to action to these different stakeholders, whether it's just to mitigate acidification or to invest in some of this work in different ways. So I kind of repeated that, so I don't think you need to know more about that. I'll just give you a quick example. And then the link that Kirsten will send around has our new little landing page where you can find the developed Alaska story map, although all the other regions are still sort of behind the veil and we'll be releasing them slowly this month. So here's just an example of Alaska. We decided to begin each story map with a flagship species that we selected. This was a fun opportunity for us to move beyond shellfish, and I say that with a hug to myself because I am always focused on the shellfish, and it was an opportunity for me to say, all right, this is a broad storytelling opportunity. This is just placing acidification in the context of climate change, in the context of ecosystem change, and we don't need to be, you know, in this particular case, in some cases we do,
But in this particular case, we don't need to be so tethered to having a specific biological impact study that we're pointing to, and we can talk in broad terms. So we picked the puffin for Alaska. So we say, hello, I'm a horned puffin, welcome to my community. You've probably seen me doing these things. And unfortunately, my home and food source are under threat because of climate change. And that's sort of the structure that follows a lot of these flagship species. I won't walk you through the whole thing, but of course we did get the pH scale in there. We are trying to be a little bit nuanced as we walk through introductions of acidification, and then also introductions to the whole picture, right? Thinking about these other stressors that are combining to have impacts to habitats and species availability and location. And then we go into cute little sections that are really trying to synthesize and condense a lot of information going on in one location. So Alaska's waters are both cold and old. Small changes can have big impacts on species. And each region sort of has that little hook that is somewhat repeats itself in the different regions, but has details per region to sort of illuminate what's happening there that is, again, particular to the area. Of course, we have a whole section on impacts to humans. So we've got quotes about different communities. We have different locations on the map showing the activities going on in the region, including and mostly curated through the cans. And then here's an example of our calls to action. So we have 4 total. Here's just 2 little examples. Using your voice, vote for the ocean, which is a campaign that the aquarium have queued up. Waving goodbye to carbon and really getting into a carbon forward narrative about efforts to reduce carbon are tied to mitigating acidification, and we go on from there. So just very quickly about what we are hoping to accomplish. I mean, it's very simple stuff. So the first one is just increasing understanding about the impacts of carbon on our ocean. That is still a gap too far for many people, as you all know, including very educated, very policy-forward people, very ocean-forward people. And so that's just sort of square one, is introducing carbon and climate change as a problem for our oceans and establishing OA centrally in that problem. A big one for me is reaching new audiences. You know, I think in our community of OA practitioners, if you will, we're really generating information to engage our stakeholders in pretty discrete purposes, as we've heard about and we all engage in. For me, this is just, you know, we always sort of say, it's not the general public. You don't want to be, there is no such thing as a general public, you know, don't tailor your work for that. But in this case, it was like, no, the general public is sort of embracing that particular constituency of U.S. aquaria or marine learning centers. And then, as I mentioned, the third kind of exciting element for me was really thinking about what is the call to action for a general public and putting that on paper, and that by doing that, we are really, in one way, starting to be part of this effort of moving the narrative from OA science to action taking across different constituencies. So again, we've talked about that. I'll just only say that in addition to visitors just coming through, looking at displays, engaging with what's in an aquaria or marine learning centers, we've increasingly realized that a lot of the aquaria are involved in school programs, curricular development, community or nonprofit groups. I mean, you've probably worked with aquaria in the area, including right down the street. And so there's a lot of sort of secondary audience that aquaria are reaching, and this is one tool that we can have, that they can be utilizing in that bucket of outreach. And then across the story maps, as you'll see, we actually have call-outs for things that the federal government are doing, things like national parks, tribal governments, city governments. And so you could really imagine that those constituencies also could use this in outreach, engagement, link it on a website, things like that. So that's what we're hoping to have a little bit of an inclusive content enough that they see themselves in the work and want to share out. And then here's just a quick little idea again of we're in sort of like phase 1 of just getting the story maps developed on this landing site that you will see. That landing site will also be somewhat replicated for NOAA OAP as well as the Aquaria Conservation Partnership. So all 3 of our partners have all the story map collections on their homepage. But phase 2 is going to look a little bit more like this, which is one-on-one, not hand-holding, but reaching out to each aquaria, particularly to sort of better understand what do their displays look like, what does their technology look like, flyers, banners, stickers, codes, websites, address, all those types of things, and really thinking out individual curation needs for those that are really interested and want to be digging in. And just a sense of timeline and expectation, by the end of October, certainly, probably before, all of our story maps will be available and live for you all to utilize and think about and share out. Then we will be developing some how-to guidance, particularly for the CAN partners as well. I think one thing we realized quickly was that, that we really want the Aquaria to be using this, but the CANs have loads of locations that you are all involved in that have visitor traffic, where this type of tool or outreach material might be shared. So we really want to get a strong sense of how-to guidance, process these for requesting changes or individualizing something you think would make it extra special for where you want to showcase it, and make sure that that type of dynamic nature is available, but also that there's some boundaries in place so that we're not making a zillion iterations. And then, like I said, phase 2, working with the Aquaria one-on-one, will probably last to the beginning of next year, and really making sure we have some committed, high-trafficked Aquaria that have some meaningful content that we can really point to as a big success here. So there's our landing page. Again, Kirsten sent it around, so it is recently developed by me, meaning it is not perfect and beautiful. Someday it will be when our graphic design person returns from getting married. But this is the very rudimentary version. And so it's only going up from here, but definitely encourage you to check it out. Reach out if you want to learn more, or someday share this out at a different venue or with certain partners. And I'll just wait for questions to share any other insights. How's that? Okay, we won't spend a ton of time on questions so that we can just kind of keep the afternoon moving and not hold you too far past 5 o'clock. Do we have any questions for Jesse, Emily, or Jen? Okay. That's okay, you guys can talk to them at the coffee break, and I will just say that it is 100% on me that the SOCAN one hasn't gotten done yet. So I will be on that next week.
No, I'll quickly say, this has been, I should clarify that the CAN's individual leads have been the ones, there's a woman, Edie from Alaska, that's really been driving this process, but the CANs have been the leaders of curating some of the content back to us to plug into these different local aspects, and it's just been such a fun collaboration with the CAN community, so we are super grateful for the opportunity to work with you guys in this way. Thank you.
Why do you have this pleasure guy? Yes. It's like it goes out and do like a target.
All right, so we'd like to get going
You use it to try to test
on the last section for today.
Let's go around somewhere.
So in this section, we're actually going to cover federal and state perspectives and considerations, not just on what's going on with OA, but we're going to branch out a little bit and cover some DEIJA initiatives. New York State Department of State is providing us today with a short video on DEIJA. And unfortunately, even though they were supposed to be here today, it didn't quite work out, but thank you very much for supplying us with this video.
Good afternoon. Hello, everyone. Great to see you all here, and we regret we couldn't be there in person. My name is Nicole Jean Christian. I am the Department of State Diversity, Equity, and Inclusion Officer and Senior Policy Advisor to the Deputy Secretary for the Office of Planning, Development, and Community Infrastructure. Today I'm joined with two of my colleagues who are members of our DEI-JA working group, Lizzie Mogus Garcia, who's a NOAA Coastal Management Fellow, and Jeff Herder, who's the Division Director for the Office of Planning, Development, and Community Infrastructure. So today what we'd like to do in the few minutes that we have is discuss ways that we can more effectively engage groups that we want to reach and enhance participation levels in processes, decision-making, and discussions about ocean acidification. We want to work together with communities to develop solutions and to discuss issues and to create possible remedies. Next slide, please. While on a preparation call a few weeks back, Jeff and Christy and Janet and I were discussing how this conversation started and where we could go from here. An important highlight was how the science community and its vision has expanded even more in the face of climate change. We speak to each other, we speak to communities, and we speak more often and more frequently because of the impacts of climate change. We have a collective need and desire to connect with impacted communities and populations more than ever before. Important considerations for us include considering the impact of ocean acidification on communities that have less resources or are under capacity, and understanding how ocean acidification affects the local economy and those communities that we target as the next frontier in the blue economy. As we know, new jobs will be created as a result of some of the changes we see, and we want to support and enhance our coastal community economies and ecosystems. And it starts with how we reach the non-science community. Next slide, please.
Good afternoon, my name is Jeff Herder, and I'm the Special Projects Manager with the Office of Planning, Development, and Community Infrastructure. So my background is from the science side of things, and so I want to speak to you about the things that you recognize, the effects that we've been working on, and the ripples of a healthy ecosystem, and healthy shellfish populations. So you all recognize that ocean acidification can impact the shellfish population, the opposite from the diagram that we're exhibiting on this slide. So healthy shellfish population leads to healthy seagrass populations and healthy estuaries and fish breeding areas, especially commercially important fish species. And that drives the economies of the area, and so they're all interconnected. And while the communities we Eastern Coast coastal areas may be experiencing these effects, they may not realize they're as a result of coastal acidification. Next slide, please.
One of the first steps is for us to reframe how we think about our value added to impacted communities. Ocean acidification has rippling effects and impacts across our lives, and it's important for us to be able to speak about the impact in less technical terms to non-science communities. To do this, it can help to first start with being clear about what we want from the community. Are we providing tools? Are we giving them planning assistance? Are we helping them with preparation? Are we supporting them in their work already? Are we building capacity? What is it exactly that we are providing, and what do we want from the community? Enhancing public participation and increasing engagement is a hallmark of equity. One of the most effective ways to enhance both participation and engagement is to be very clear about the value of your activity, your event or process. Impacted communities are juggling many priorities. How can what we are offering fit those priorities and rise to the top? By being clear about the value and what is being provided, we can better position ourselves as partners. And partnership is the beginning of building relationships that will go beyond the immediate project or process cycle.
Okay, begin by considering the words we use. How we refer to the non-scientist community and impact communities. Different words have different meanings for different people. The word stakeholder in this definition dates back to 1708 when it was a term to mean a holder of a wage. It referred to a wager or gambling activity where each person put into the holder's purse an equal portion of the bet to hedge their loss, bet or stake. As time evolved, this term adopted from a purely business perspective usually referred to a business or wager transaction. Eventually, the term evolved and was adopted by the business community in general, referred to someone who has an interest or concern in something. As time passed, the term stakeholder was used generally to refer to groups of people or populations of people with a shared interest. The term, some would present, has been used so broadly that at times the unique characteristics, experience, or needs of a group are lumped together. There are some terms that are better suited to fit today's evolving diversity of people, populations, needs, and experiences. In light of our renewed community to commitment to equity, seeking to identify, mitigate, and remove, where possible, barriers to program access, there are other terms more frequently used to refer to our community partners. It truly reflects our evolving relationship with them. Next slide, please.
So hello everyone, my name is Lizzie Mogus Garcia and I'm a 2022 to 2024 NOAA Coastal Management Fellow here at the Department of State, working in the Climate Change and Resiliency Unit as well as working with the DEIJA working group. So talking more about the words that can re-traumatize when we're talking in terms of stakeholder. Continuing to use a term that has a negative colonial or origin or context can re-traumatize participants and could be considered disrespectful, causing mistrust in some cases. The issue with the word stakeholder is that in a colonial context, a stakeholder was the person who drove a stake into the land to demarcate the land he or she was occupying or stealing from indigenous territories. So the continued use of the term can be constructed as disrespectful of indigenous people, as well as perpetuating colonialization and re-traumatization. So this term, stakeholder, may no longer be an accurate description to describe the community's interest that we work with. The discussion today is sharing that we have learned about the term and its continued use. So we have alternatives to share, and we will start this discussion and evolve strategies and language towards shared design through enhanced public participation, engagement, and partnership. Next slide, please. So, speaking of those alternatives, we have some great alternatives to the term stakeholder highlighted here. Some of those include co-designer, participant, contributor, impact creator, influencer, or simply those that are in the circle of engagement or those who care. So all of these terms in some cases more accurately describe how we seek to engage communities today. And so with a focus on equity, as well as addressing the causes of inequality or barriers to access, we evolve in how we refer to and conceptualize communities, people, and solutions. So we acknowledge that our actions impact others and that we benefit greatly from their involvement and their value in the process as a requisite to meeting our goals, and then therefore seek to shared problem solving and shared decision making. The communities we work with and the role we play now together is partnership, and with that, this newer language that we're learning can better reflect this evolving experience. So thank you very much, and next slide please.
So how do we create opportunity for engaging communities in co-designing, participating, and contributing? We're so glad you asked. So as we dive deeper into this concept of co-design and moving from stakeholder to contributor, we start to think about how to relate to impact communities, partners, co-designers, and collaborators. On the slide, we have an infographic that was sent to us by fellow work group member Christine Mansfield-Morgan. It shows a figure or image that a lot of us have seen that illustrates the difference between equality and equity. But this is a new version that includes justice. So on the left, we see equality as everyone benefiting from the same support. So they each have one box to stand on. In the middle, we have equity as individuals are given different supports based on their unique experience or either the experience of need. Boxes to stand on differ based on ability to see over the fence. Finally, on the right, we see justice. All three can see the game without any supports or accommodations because the cause of the inequity was addressed or the systemic barrier was taken down. We create opportunities for participation by reframing the discussion to think about our value to the greater experience of justice. An open system where everyone benefits systemically, procedurally, and long-term. Next slide, please.
What is the difference between public participation and engagement? What do you think they mean? This is so important since this field is rapidly evolving, and as we learn, we share with each other like we are today. Sometimes public participation and engagement or community engagement are used interchangeably, when in fact they are quite different. Public participation is quantifiable. These are, as they say, cheeks and seats, the actual folks that attend your activity or event. The results can be counted. Public engagement are the event types or opportunities that you provide. So these are the activities you create to attract your audience. Public engagement that is co-designed or nested within an already existing community structure has a higher probability for traction and long-term participation. When we partner with communities in co-design to offer activities, programs, and processes, we broker or create the possibility for longer-term traction. Take a look at this quick exercise. We know school started this week, so this is a take-home exercise for homework and an opportunity for you all to discuss later. On the left, you see public participation. On the right, you see the circle of public engagement. The intersecting area in the middle is where we can co-design. That's the opportunity to share. So take out a piece of paper when you have a chance, draw two interlocking circles, put PP on one side and PE on the other. Both are important and are relying on each other for long-term success. The area in the middle where both circles intersect is an area of opportunity where you can co-create, share design, or nest activities in an existing program structure or schedule. Think about a time where you attended an event with a high level of attendance, and when you attended a uniquely organized event. Jot your thoughts in the circle on each side. For public participation, jot your thoughts about how many people were there and what they were tasked with doing. What activities did they complete? Then on the right side, jot some of your thoughts about the types of events that you've seen work. In the middle, you can fill in over the next couple of days or together in this exercise this afternoon, ideas and ways that you think you can combine the best of both. High attendance with an innovative activity or event. So just think about how you can combine the best of both and how you can create something new. What is it that you know? What is it that you know now that actually works that you can share? Thank you all for your time.
All right, for the remainder of this session, we have all live speakers. So at the end of this session, we're going to have more of a panel discussion. So we'll ask panelists to come up to the table after the full session or whenever they feel comfortable. So we're going to start with Courtney Bukowski, who is representing the Interagency Working Group on Ocean Acidification. Then we have Casey Personius from New York's DEC, Megan Rukowski From New Jersey's DEP, Molly Yanko. From DNREC, or Delaware Natural Resources Environmental Control, thank you. And then Rachel Lamb from Maryland's DOE. And then we'll have the panel discussion after.
All right. Hi, everyone. I'm Courtney Wachowski. I am with NOAA's Ocean Acidification Program, and today I'll be speaking to you on the interagency working group on ocean acidification. You've heard it referenced a few times already as the IWGOA. I'll try not to use the acronym too much. And I'll be sharing about how our group implements OA policy at a federal level. A short background for those of you who aren't familiar with the working group. It was established by legislation in 2009, really for the main purpose of coordinating federal activities around all things ocean and coastal acidification, from monitoring, research, to education and outreach. We have members that represent 14 different federal U.S. agencies. The group is chaired by NOAA, and I serve as the executive secretary. I want to note that we have a really diverse group of agencies who all engage with OA in different ways, influenced by their unique missions and mandates. And one of the main roles of the interagency working group, I'm sorry, can I get a quick glass of water? Okay, sorry, I'm going to start that over again. So one of the main roles of the IWG is to decide for, the nation at the national level, what should be our priorities for OA actions, and how do we best coordinate those activities among federal agencies. And one of the main ways this is codified is through our strategic research plan, which is the first project I want to give you an overview of. And the first plan came out in 2014, and I'm excited to say the second edition is expected to be released later this month. And so I'll be sharing that with Janet and encourage you all to take a look at it when it comes out.